In the bustling metropolis of a cell, energy is paramount. Just as cities thrive on a constant supply of power, cells require a steady stream of energy to fuel their myriad functions. Enter the mitochondria, often hailed as the powerhouses of the cell. These remarkable organelles, with their intricate structure and elegant mechanisms, stand as testament to the awe-inspiring complexity of life at the microscopic level. Imagine them as bustling factories, diligently working to produce the energy currency of the cell, ATP, or adenosine triphosphate. Mitochondria, unlike many other cellular components, possess their own DNA, a relic of their ancient origins as free-living bacteria. This unique characteristic underscores their vital role in cellular respiration, the process by which cells convert nutrients into usable energy. Within the mitochondria's double membrane, a symphony of molecular events unfolds, culminating in the production of ATP, the molecule that powers countless cellular processes, from muscle contraction to nerve impulse transmission. The process by which mitochondria generate ATP is known as oxidative phosphorylation, a term that hints at the elegance and complexity of this intricate metabolic pathway. It's a process that has captivated scientists for decades, and its intricacies continue to be unraveled, revealing the breathtaking ingenuity of nature's design. Oxidative phosphorylation, the driving force behind ATP production, can be likened to a carefully orchestrated waterfall. Imagine a series of cascades, each representing a step in this intricate process. As water flows down the waterfall, losing potential energy with each drop, electrons are shuttled along a chain of protein complexes embedded within the mitochondrial membrane. This chain, known as the electron transport chain, is the heart of oxidative phosphorylation. The energy released as electrons cascade down this chain is harnessed to pump protons, tiny positively charged particles, across the inner mitochondrial membrane. This creates a proton gradient, a difference in proton concentration across the membrane, akin to water piling up behind a dam. Just as the water behind a dam holds immense potential energy, this proton gradient stores the energy needed to drive ATP synthesis. The controlled movement of protons back across the membrane through a molecular turbine known as ATP synthase provides the mechanical energy to generate ATP from its precursors, ADP and inorganic phosphate. This remarkable process, known as chemiosmosis, couples the flow of protons down their concentration gradient to the synthesis of ATP, the cell's primary energy currency. Central to the process of oxidative phosphorylation is the electron transport chain, a remarkable example of molecular teamwork. Picture a relay race, with electrons as the baton being passed from one runner to the next. In this cellular relay race, the runners are a series of protein complexes embedded within the inner mitochondrial membrane. The first runners in this relay are NADH and FAD2, molecules laden with energy-rich electrons derived from the breakdown of food molecules. They arrive at the starting line, eager to offload their precious cargo. As they pass their electrons to the first complex in the chain, NADH dehydrogenase, energy is released, propelling protons across the membrane. The electrons, now carrying slightly less energy, are passed along the chain to subsequent complexes, each transfer releasing a burst of energy used to pump more protons across the membrane. Finally, the electrons reach the last runner in the relay, cytochrome C oxidase, where they are accepted by oxygen, the final electron acceptor, forming water. In the intricate ballet of oxidative phosphorylation, NADH and FAD2 take center stage as the star players. These two molecules, often overshadowed by the more familiar ATP, play a crucial role in initiating the electron transport chain. Think of them as dedicated delivery trucks, ferrying precious cargo, energy-rich electrons, from the bustling metabolic pathways of the cell to the mitochondrial membrane. NADH, or nicotinamide adenine dinucleotide, is a coenzyme derived from vitamin B3, while FAD2, or flavin adenine dinucleotide, is derived from vitamin B2. Both molecules are essential for cellular respiration, acting as electron carriers, shuttling electrons from the breakdown of glucose and other fuel sources to the electron transport chain. As these molecular trucks offload their cargo at the electron transport chain, they are recycled back to their oxidized forms, NAD+ and FAD, ready to pick up another load of electrons. This continuous cycle of reduction, gaining electrons, and oxidation, losing electrons, is essential for maintaining the flow of electrons through the electron transport chain, and ultimately for the production of ATP. Now, let's dive into the fascinating world of molecular turbines. You see, ATP synthase is like a tiny motor within our cells. It's amazing how this enzyme works. It spins at an incredible speed, converting ADP and phosphate into ATP. This process is crucial for powering almost every cellular activity. Think of it as a turbine generating energy, but on a microscopic scale. Isn't it incredible how such a tiny structure can have such a massive impact on our lives? This really highlights the intricate and beautiful complexity of life at the molecular level. Imagine, if you will, a dam holding back a river. In our cells, we have a similar structure, the inner mitochondrial membrane. This membrane acts like a dam, maintaining a gradient of protons. It's this gradient that drives the synthesis of ATP, the energy currency of the cell. Just like water flowing through a turbine generates electricity, protons flowing through ATP synthase generate ATP. It's a remarkable process, really. But this process isn't always foolproof. Sometimes things can go awry. The proton gradient might dissipate, or ATP synthase might not function optimally. When this happens, the cell's energy production can be compromised. It's like a dam with leaks. The energy production becomes inefficient, and the cell struggles to meet its energy demands. When our cellular powerhouses, the mitochondria, falter, the consequences can be significant. Cells might not get the energy they need to perform vital functions. In extreme cases, this can lead to cell death. It's like a power outage in a city. Without electricity, everything comes to a halt. Similarly, without ATP, cellular activities cease and the cell's health is jeopardized. Despite these challenges, the production of ATP is a symphony of energy, a beautifully orchestrated process. Each component, from the electron transport chain to ATP synthase, plays a crucial role. It's this intricate dance that sustains life, providing the energy needed for every heartbeat, every thought, and every movement. Truly, it's a testament to the elegance and efficiency of biological systems.